Hello everyone. Hope all are doing good. It's been a long time since I met you all through a video. So now Agam webinars are taken another initiative of including embryology videos. Now I hope you all know embryology is part of first year anatomy. Yes. See, from university point of view, in paper 1 you can expect 2 or maximum 3 embryology questions, and paper 2 again 2 or 3 questions. And if you get any viscera pertaining, if you get any essay question on viscera, then its development can be part of an essay question. So from exam point of view, it's not that important. It is not going to give you maximum amount of marks. But trust me, if you're not going to study embryology in first year, you will not be able to understand systemic pathology in second year. You will not be able to understand any congenital disorder pertaining to clinical subjects like ENT, optal, pediatrics, surgery. Pediatrics definitely is going to revolve only on embryology. So if you know embryology, abnormalities in the development is what we are going to study in pediatrics. So embryology is very, very, very important. And we usually neglect embryology, so which nobody should do. So what I have planned is something called embryology series. So we will have 3 videos per week in the morning on Monday, Wednesday and Friday on embryology, and all the videos will be taken by me. I'm going to deal the entire embryology for you all. So it will be on 3 days on Monday, Wednesday, Friday at 11 a.m. And what I want you all to do is to sincerely follow the embryology videos. At least if you're not going to spend time with Langman or other embryology textbooks given by your college, but please listen to the videos. So videos will not exceed 25 minutes. The maximum time limit that I have set is 20 to 25 minutes. I will not take even 1 second more than that. Maybe this video, since I have given an introduction, I might consume time, but even that is not confirmed, right? So please kindly watch the embryology videos that will be put regularly on the mentioned days. Right now, embryology, we have got 2 branches, general embryology and systemic embryology. So we will be starting with general embryology and then we'll move on to systemic embryology. So the topic for today's class is on gametogenesis. So our embryology will start with understanding how the gametes are formed, when the gametes are formed, and how it is going to act, right? Now, let us get started. So if you take gametes, yes, they are going to arise from a group of cells called PGC, that is primordial germ cells, right? PGCs. Now, what is the origin of this cell? A cell cannot suddenly come, right? So this primordial germ cell are going to arise from a layer called epiblastic layer of the embryo at the second week of the intrauterine life. So when we study about the second week development, we'll be talking about the epiblast, hypoblast, etc. So from the epiblast layer at about the second week, we have got some cells called primordial germ cells that arises. So this is what is going to give rise to gamete. So this primordial germ cell is a pluripotent cell. Please note that word, pluripotent stem cells. So from the primordial germ cell, that is from the epiblast layer, this primordial germ cells will migrate to yolk sac. So yolk sac will be developed around the end of 3rd week or beginning of 4th week. So it migrates to the embryo sac, it migrates to the yolk sac at about 4th week. Yes, at about the 4th week of intrauterine life, it migrates to the yolk sac. And from the yolk sac, ultimately gamete should reach where? Yes, it should reach the gonads, right? And it reaches from the yolk sac, it reaches the gonads at about 5th week. Right, about the 5th week. So this primordial germ cell, once it reaches the gonad, it is going to give rise to gametes. So we are going to see how the gametes are formed. So primordial germ cell, which is a pluripotent cell, which arises from the epiblast layer at the 2nd week, it migrates to the yolk sac at the 4th week, reaches the gonads at the 5th week. So all these can be MCQ's question. Now, if you look at this picture, yes, so can you appreciate primordial germ cells in the wall of yolk sac? So this is the yolk sac. In the endodermal layer of the yolk sac, you will have primordial germ cells at about the 4th week. From there, the cells will migrate to the gonads at about 5th week. About 5th week. Right? So this is about the initial aspects, right? Primordial germ cells. Now, what kind of stem cells they are? Yes, they are pluripotent stem cells. Yes. Now, why should I know about primordial germ cells? Why didn't I directly jump into gametogenesis? Is because primordial germ cells, they have to migrate to gonads, right? So from the yolk sac, they have to get to gonads. But instead of that, these cells can migrate upwards and it can reach the oropharyngeal aspect of the embryo. If it reaches the oropharyngeal aspect, generally these kind of abnormal migration can lead on to a tumor called teratoma. Now, we will be studying about teratoma in systemic pathology in second year. What is this teratoma? Is basically tumor of germ cell layers. Yes, it is tumor of germ cell layer. So we all know we have got ectoderm, endoderm and mesoderm, right? So this teratoma is a tumor of germ cell layer. So it can contain tumor of epidermal cells or mesodermal cells or endodermal cells. So usually this teratoma will contain lots of components like skin, hair, muscular tissues and etc. Right? So since the primordial germ cells are pluripotent, they can give rise to many kinds of cells. They can give rise to epidermal cells, so the teratoma will contain epidermal content. They can give rise to mesodermal cells, so we can have mesodermal content. So it is a germ cell layer tumor. This is what you have to know. Why there is teratoma? Because of primordial germ cells pluripotency. Right? Yes, this is enough. Now let us get on to gametogenesis. So first what we are going to learn is about oogenesis. So what is oogenesis? Is formation of female gamete, right? So primordial germ cells, right? So at the 5th week will reach the ovary, right? Or the female gonads. At what time? Yes, 5th week. Once the primordial germ cells reach the ovary at the 5th week, what will happen is these primordial germ cells will undergo mitotic division to form oogonium. Yes, it will undergo mitotic divisions to form what is called as oogonium. So whatever primordial germ cells have reached the gonads will form oogonium at about 3rd month of intrauterine life. Yes, whatever duration I'm mentioning is a very important MCQ question. So oogonium will be formed at 3rd month, right? Now this oogonium in turn will undergo another set of mitotic division to form what is called as primary oocyte, right? It will undergo meiotic divisions to form what is called as primary oocyte, right? So this oogonium will undergo mitotic divisions. It will undergo mitotic divisions again to form primary oocyte. So oogonium that has been formed in the 3rd month will undergo mitotic division to form primary oocyte. So this will take approximately 5 to 7 months. Yes, it will approximately take 5 to 7 months. Now, next, this primary oocyte will enter into meiotic division. Yes, it will enter into meiotic division. But what happens is, it will not complete its meiotic division. Yes, it will not complete its meiotic division. Instead, what will happen is, it gets arrested. Yes, it gets arrested at prophase 1 of meiosis 1. Yes, prophase 1 of meiosis. Very important MCQ. Yes, at the prophase 1. So prophase 1 has got 5 stages. At what stage of prophase? Yes, it is diplotin stage. So once by the 7th month, all the primary oocytes have been formed, it gets into meiotic division, it gets arrested at the diplotin stage of prophase 1. And this oocyte which gets arrested at the diplotin stage will remain in the arrested stage till puberty. Yes, it will remain in the arrested stage till puberty. So this kind of a structure which is present in the female baby till puberty, that I refer to as primordial follicle. What I refer to as, yes, it is primordial follicle. Now, if you appreciate this picture, can you make out? So this is the developing oocyte, and surrounding it, I have got a layer of epithelial cells. It is a flattened epithelial cells, very important. It is called follicular cell. So oocyte with a layer of flattened epithelial cell is called as primordial follicles. So till puberty, the ovary is going to contain primordial follicles. It is going to contain primordial follicles. Now, if you take at birth, the number of primordial follicles will be in lakhs, approximately 6 lakhs to 8 lakhs. But when it comes to puberty, we will be having only around approximately 40,000 primordial follicles, right? At birth, it will be around in lakhs. And when it comes to puberty, it will be only around 40,000. Now, the female has attained puberty. So what will happen is this primordial follicle, right? It will enter into ovulatory cycle. So you have all the hormonal influences that happens when the female is slowly reaching a puberty. So when that happens, the follicular cells that are present in the primordial follicle will start growing. Can you make out? Initially, it is a flattened cell, but what happens following the hormonal stimulation? It becomes a cuboidal cell. So the follicle that is surrounding the oocyte will start growing, right? So if you take the flattened epithelial cell, it becomes a cuboidal follicular cell. At the same time, can you make out a red color structure here? Is that is called 𝗂𝗵𝒂𝗃𝗃. So what happens is at one point, what is happening is the flattened epithelial cells are getting converted to cuboidal follicular cells, and these cuboidal follicular cells, listen carefully, the cuboidal follicular cells and the cytoplasm, that is the oocyte cytoplasm, it secretes certain substances, right? So from the follicular cell, you have got some substances. From the cytoplasm of the oocyte, you have got certain substances. Those combine to form a layer called as 𝗂𝗵𝒂𝗃𝗃. So the follicular cells will keep expanding on one side, it will grow in number. At the same time, there is development of 𝗂𝗵𝒂𝗃𝗃. So this is what is called primary follicle. So primary follicle is basically primary oocyte with 𝗂𝗵𝒂𝗃𝗃 with layers of follicular cells. Now, what will happen to this primary follicle? So there is hormonal stimulation that is happening. The primary follicle, right, will undergo further development. And what happens is at one point of time, there is formation of a cavity. Yes, the follicular cells that are present as a group will start arranging themselves, leaving behind a cavity called as antrum. This antrum is a fluid-filled cavity, right? So this fluid keeps expanding. Yes, it keeps expanding, and finally it gives rise to a very big cavity called as antrum with the external layers called theca interna, theca externa, and you have got the oocyte here. So this is called as Gsraphian follicle. Yes, this is called as Gsraphian follicle, right? So this Gsraphian follicle, which is filled with antrum, it has got a theca layer, that is, it is divided into theca interna and theca externa. What is this theca basically? Is these granulosa cells, the follicular cells, which is going to line the oocyte. So can you appreciate, this is the ovarian stroma. Yes, this is the ovarian stroma, this is the oocyte. So the follicular cells, basically they separate the oocyte from the stroma. So these follicular cells will in turn get arranged into two layers, that is theca externa and theca interna. What is 𝗂𝗵𝒂𝗃𝗃? Yes, that is the one that lines the oocyte, right? Developing oocyte, which is formed from the secretions of the follicular cell and the oocyte, right? So this Gsraphian follicle. This Gsraphian follicle on LH search will complete its first meiotic division. It will complete its first meiotic division to form secondary oocyte and a polar body. Yes, to form secondary oocyte and the first polar body. So primordial follicle, primary follicle, then you have got secondary follicle, tertiary follicle. Finally, I've got a Gsraphian follicle with the antrum. And this on LH search, it completes its first meiotic division to form secondary oocyte and the first polar body. Yes. So let me erase all the ink on the slide and let me have only secondary oocyte and first polar body. Now, this secondary oocyte, again a very important question, will enter into meiosis 2. Yes, will enter into meiosis 2 and get arrested at metaphase of meiosis 2. Yes, It will get arrested at metaphase of meiosis II. So it will wait for 24 hours. Okay, it will wait for 24 hours. So within 24 hours, if the sperm comes, fertilization takes place. So meiosis II will be complete. Okay. Meiosis II is complete. If the sperm is not going to come, then the corpus luteum that is formed. I hope you all know what is corpus luteum. The secondary oocyte, right, which is formed as a result of first meiotic division, will rupture all the granulosa cells and come out. So the ruptured granulosa cells are called as corpus luteum. Right. So if this secondary oocyte is not formed, right, if it is not fertilized by the sperm, the corpus luteum will get converted to a scar tissue called as corpus albicans. Now you might ask, what is the significance? Yes, corpus luteum is necessary for maintenance of pregnancy because that will be the source of progesterone initially, right? So if it has been fertilized by the sperm, yes, meiosis II will be complete and corpus luteum is needed for progesterone supply. If it is not fertilized, this corpus luteum within approximately 9 days will get converted to corpus albicans, leaving a scar, and bleeding will happen. That is menstruation, right? So this is about oogenesis, right? I hope you all got. So what is very important is what happens to primary follicle gets arrested at the diploiding stage of phase 1. What happens to secondary oocyte gets arrested at the metaphase of meiosis II. It will wait for 24 hours. If within 24 hours sperm is going to hit the ova, yes, meiosis II is completed, corpus luteum is going to be there. If it is not going to happen, within 24 hours the sperm is not going to come, it will get converted to corpus albicans and it will proceed for menstruation, right? So that is the story, right? Now coming on to spermatogenesis. So very simple, spermatogenesis is very simple. Oogenesis, it began right from birth, right? But spermatogenesis will begin only at puberty. Very important point. Second difference between spermatogenesis and oogenesis is, if you take both the first meiotic division and even the second meiotic division will lead on to formation of ova. You will get the formation of female pronucleus and you will have secondary polar body. We'll be seeing in fertilization in the next video. So again, if you take, you have got formation of polar bodies in spermatogenesis, but here you will not have any polar body formation. Right? These are the two things that you have to know. Now, again, you have got primordial germ cells, right? So if you take till puberty, this primordial germ cells will remain as primordial germ cells only, right? Where it is going to stay is it is going to stay in the immature sex cords of the testis. So if you take, suppose this is the sex cord, the primordial germ cells are going to be there in the sex cord, which is surrounded by certain supportive cells, which in turn is surrounded by certain supportive cells. So when the boy attains puberty, what happens to the sex cord is the sex cord becomes the seminiferous tubules. Yes, it becomes the seminiferous tubules. And the surrounding supportive cells that we saw, it becomes the Sertoli cell. So again, it is meant for nutrition, right? So the sex cords becomes the seminiferous tubules and the supportive cells becomes the Sertoli cells. So the spermatogenesis, where it is going to happen, is it happens in the seminiferous tubules, right? Now let us see the process of the spermatogenesis. Is so at puberty, the primordial germ cell will undergo mitotic division to form spermatogonia. Yes, this spermatogonia will undergo another mitotic division to form primary spermatocyte. Yes, it will undergo another mitotic division to form primary spermatocyte. This primary spermatocyte will undergo meiotic division. As it undergoes meiotic division to form secondary spermatocyte. This secondary spermatocyte in turn will undergo another meiotic division to form spermatids. So spermatids, initially they are not the matured sperm. So this spermatids will undergo certain changes to form the spermatozoa. Will undergo certain changes to form spermatozoa. So if you take primary spermatocyte is formed from spermatogonia as a result of mitotic division. The primary spermatocyte gets converted to secondary spermatocyte by means of meiotic division. The secondary spermatocyte undergoes another meiotic division to form spermatids. And very important is the spermatids gets converted to spermatozoa by a process called spermiogenesis. Genesis means generation, right? So from these spermatids, I've got generation of spermatozoa, hence the term spermiogenesis. Now you all know meiosis is a reduction division. So one parent cell will give rise to two daughter cells, right? So if you take one spermatogonia, let me write it in another color, running out of time. One spermatogonia, mitotic division, I've got one primary spermatocyte. It undergoes meiotic division to form two secondary spermatocyte. These two secondary spermatocytes in turn undergo meiotic division to form four spermatids. And these four spermatids undergo spermiogenesis to form four spermatozoa. So ultimately, if you take one spermatogonia, it undergoes spermatogenesis to form four spermatozoa. And very important NEET PG question is, it takes approximately around 74 days for the completion of spermatogenesis. Very important. It can be asked even in your MCQs and university exam. It takes around 74 days to form spermatogenesis. Right, this much is enough. Our book says you have got type A spermatogonia, type B spermatogonia, and if you take this spermatogonia, which is formed as a result of mitotic division from primordial germ cell, it undergoes 5 to 7 mitotic divisions. So what will happen is you will have 5 to 7 mitotic divisions, right? So you'll be having approximately 32 to 64 spermatogonia. So you will be approximately having 64. You will be approximately having 64 to 256 spermatozoa. This is given, but you need not confuse, right? What is the basic concept is, this primordial germ cell will undergo 5 to 7 cycles of mitotic division to produce multiple spermatogonia. Here, what I have considered is only one mitotic division, right? So that is the thing here. So it takes approximately 74 days. You have to know this. And one spermatogonia will give rise to 4 spermatozoa. This is the take home point. Right, so if you take type B spermatogonium, you have got primary spermatocyte, secondary spermatocyte, spermatid, which undergoes spermiogenesis to form spermatozoa. So what is this spermiogenesis? So what happens exactly is, you have three things. One is formation of acrosome. As you all know, a sperm will contain acrosome, then you will have neck, body and tail. Right, so you have formation of acrosome, then there is condensation of nucleus, then there is formation of neck, middle piece and the tail. So there is complete maturation of a structure. So if you take, this is the spermatid, so you have formation of acrosome, then the remaining parts like the neck, body, tail, along with the formation of nucleus. Right, so this is what is called spermiogenesis. Right, so this is about the gametogenesis. Right, a very quick recap I have given. So with this, thank you all. We will meet in the next video. Tata, bye, take care.